morning. I'm going to do something interesting or possibly dangerous. My notes are here. That's K-trick, and it's editable. It's in Overleaf, it's in LaTeX, but there's also like a rich text interface. So if there are questions, just go into there and drop one, or if I got something written wrong there, it'll appear on my paper. It's magic. No, but if there are questions or things that I should elucidate later, I'm going to leave that up, and I'll fill it in later with more details. All right. So the kernel trick it's called a trick, but it's essentially when we have data in some space that's not easily separable, not linearly separable. So we might have two classes. Which we can't separate in a linear space. But we want to use the same tools that we're familiar with to separate them, right? Same linear classifiers and PCA or regression or something like this, where we've got a space where our data is not simple to analyze. How can we use those same familiar tools on a nonlinear space where this becomes easy? You know, for separating these two classes, or if we've got some sort of regression that doesn't follow the usual linear space. I'm not going to go into, there are a lot of sort of mathematical details for how the kernel trick is used and when it can be applied and what sort of guarantees you need on your kernels and how you get sort of sparse representations for these so that they're efficient to evaluate. I'm not going to go into those details. I'm just going to give you the sort of high-level overview of what the kernel trick is and how it works. Excuse me. There's plenty of software out there and plenty of papers out there talking about areas where it can be used, interesting kernels, and how to apply them to interesting tools or interesting problems. All right, so I'm actually going to start with linear problems and then show how we can reduce those down to just dot products, or our simple tools down to just dot products, and then show how, after we've done that, we can use kernels to lift those into a nonlinear space. All right, so we'll start with linear classification of separable data. All right. One of the classic techniques for dealing with data in this linear space, or for classification problems in a linear space, is just the Fisher's linear discriminant. So this is a very simple thing. You find well, sorry, I shouldn't say simple. This is a technique where you take the means of the two distributions. You also look at their sort of an average or sum of their covariances, and you get some vector W, which when you project upon it, separates the two data under some optimality, some loss. And that W is proportional to, for this case, sigma of the positives plus the sigma of the negatives, or sorry, the covariances and the difference of their means. Okay? I'm not going to say anything more about this. I just want to show it. It's a classic result from classification. All right, so we're just going to find a W that separates these two classes. When you project perpendicular to that, you'll get a distribution where they're hopefully reasonably separable. The details of how you calculate this you can see on Wikipedia. I'm just going to leave this aside for now and talk about a different classifier, and then we'll come back and talk about this and the other classifiers' results and how they are similar and what's interesting about them. All right. So one of the classic techniques from kernel machines or from around the kernel trick are support vector machines, which are a classification technique where, again, you have two classes. And we're going to assume they're separable for now. We'll come back to that in a little bit. And again, we want to find some direction where when we project them, they're easy to separate, or they're easy to classify along that dimension. And in this case, what we're going to do instead of using this, which was a particular form of classification error, we're going to look for the maximum margin that separates the two. We're basically going to try to find some place where when we project them, they're as far apart as possible at the extremes. So we're going to find a margin that's as big as possible. So this will be our decision boundary. This will be our margin. And this will be our projection direction. Okay? We're going to assume that they're separable for this case. So we want to make this as wide as possible. The idea being if the data is separable, we could choose any of the hyperplanes, right, and get a perfect classification error, right? We could go like this, we could go like this. But the idea behind the maximum margin is the wider we make that margin, the more confidence we have that it's going to generalize to new data, right? We've separated it as much as possible. Okay. If we look at a couple points on the boundary, actually let's use this one. Let's call this A and this B. Then the distance, the width of the margin, and we'll assume that we know this W. We're going to figure out how to solve it in a second, but we'll assume we know this W. Then our margin is equal to A minus B dotted with W, right? Just the projection divided by the norm of W, right? We're just going to take whatever length this is, take the dot product with this, and then turn this into a unit vector first. And that's how wide the margin is. All right. When we solve this, we assumed it's separable. We're going to require that when we project onto W and add some bias term, we're going to want this to be 0, this to be +1, and this to be -1. That's just how we're going to define our scoring function for the classifier. And then anything bigger than 0 or less than 0 is classified as positive or negative, and anything beyond the margin is larger than 1, so well classified. We assumed it's separable, so what we're going to require is that if this is xi, and these are all yi, their label equals -1, yi equals +1. Yep. So we're going to require, let's do this here, that for every point, its label times the output of our linear classifier, so w dot xi, so with the projection onto this W plus some bias that we'll solve for later, is greater than or equal to 1. So this says everything that's over here, the dot product of W, I should probably face W the right way, there. Everything over here, when you take the dot product of the position plus some bias, is greater than 1. And if it's negative, we just flip this, so it's less than or equal to -1. Is this part clear? We're basically going to, and then what we're going to do is make the margin as large as possible given this. Okay, so we're just going to find how we need to orient W to make that happen. When we do that, well, these are constraints, and the only other thing is we want the margin as wide as possible. We've already said that for points on the boundary, this is our width, right? So if we this right here, if we add a B, this is fixed. It's either 1 or -1 for any point on the boundary, because that's how we defined it, right? This is the width. Points on the boundary are A and B. If we add that bias, then this will be plus or -1. When we look at this part, this was constrained by being on the boundary. And so if we minimize that, minimize the width, then this margin grows. So the smaller this, given these constraints, the wider the margin. So that gives us our optimization problem. We just minimize but such that that's true. So we've got a quadratic minimization with linear constraints. Okay. So when you put that into a solver, you can you set up the Lagrangian and et cetera. Your solution is going to fall out in the form of W is equal to sum over the i's of some weights times our original i's, or original positions. Excuse me. And in fact, these alphas will only be non-zero for examples that land on the boundary. So the boundary should probably be, there should be another point here or something to constrain it. And everything else won't have any effect. And these are called the support vectors. That's where the name comes from. There's going to be some sparse set that constrain the maximum margin, depending on the dimensionality of your data. You're going to get some sparse set of terms, and then your W, your projection direction will depend only on those points that are on the margin. And so it's hopefully a small set of points. And then you can compute this, right, this is our predictor right here. Since W only depends on a few support vectors, you can compute this part of the predictor, or you can predict the output of the predictor efficiently. You only need to compare to the marginal, or the support vectors, the ones near the on the boundary. Okay. And you only really need to be able to compute dot products. So the dot product of a new point, xn, we want to predict, this is its prediction for its label. Is this greater than or less than 0? To compute this, we really only need to compute, right, the xi.xn plus some bias. So we can do this efficiently, and we only need to know dot products. Turns out we also, the part I skipped over where we actually solved it, you also only need to know dot products. And if we go all the way back to Fisher's linear discriminant, really you only need to know dot products here. You just need to take a dot product to classify some point with this classifier. You're going to dot product with this W, and that's just the mean of all the dot products of all the positive and negatives. And then some weird spatial scaling term. But what's common to all of these is we only need dot products. We only ever look at the data to solve and to project new data through this lens of dot products. And so our kernel trick is going to be, let's just replace dot products with something more powerful than linear dot products. Okay. I'm going to extend this a little bit to regression just because it's nice to see the mechanism, how it's similar. And it also generalizes this to the case where it's not linearly separable. So support vector regression with tolerance. So instead of classifying, we're going to try to regress. We have an input and output, and some function. In this case, linear, but we're going to show how we can extend that to nonlinear cases as well. We're going to set up a slightly strange formulation that you may not be familiar with, but it's useful to see if you're going to go into this space because there'll be other things like it. We're going to set up what's called new SVR. Is that new? Hope that's new. And so what we're going to do is we're going to say we want a good regressor, and we're going to define that by we're going to put similar to our maximum margin. We're going to build a regressor along these points. We want as small a tube as possible to contain them. So we want a sort of tight regressor. And we're going to be insensitive to errors less than less than epsilon. Okay, so we don't care if, as long as we have a data point that lands within the tube, no problem. All right, we're going to extend it. So here we assume linear separability, and here we're not going to assume that we can actually, that everything is clean. We're going to assume there are some outliers. Okay, we're going to handle those with an L1 error. So the distance here and the distance here, we're going to penalize with L1. So L1 for errors greater than epsilon. And so that we have some way to control how much we care about inliers and outliers. The new part is the fraction of points outside the tube is less than nu. Okay. So if we set this to 0.1, then 10% of our points will be outside the tube. So we'll find the narrowest tube, regressor tube that captures 90% of the points. Okay. Yeah.
Are we trying to find the narrowest tube and somehow simultaneously to also minimize the L1 error for the points outside the tube?
Yes. So it turns out that they combine. I couldn't find a very intuitive explanation of this. In the notes, there's a figure that talks about it where minimizing this, the way you set this up, you balance them sort of automatically. It'll find the narrowest tube that contains 90%, and then there's also maybe some regularization. So it's basically, it's not just that you minimize the L1, it's that you explicitly say, find the widest, narrowest tube that contains 90% of the points within the regressor. And what the orientation of that is, or what the particular direction function is, it will just find it. So if there's, what?
It somehow turns out that you don't have to balance those two, they don't compete with each other.
No, it's explicit. This is exactly how many will be on the boundary or outside the tube. So it also, since it's again going to be phrased as support vector machine, that's how many points you'll have to evaluate, because those are the only ones that will matter in the regression. Just like this one was defined by the points on the boundary, this one will be defined by the points on the boundary of the tube and the outliers. Okay.
So generally, like I deal with gene expressing data sets, and when you do these kind of regression, you sort of see a line, but there's a big blob at the end of line. Mm-hmm. So how would you design a confidence interval or your decision boundaries for that blob? And if you make it, how would you be very would you be very arbitrary in excluding samples or like a point from that, or like?
You don't exclude them. You just put a L1 error upon them. You can actually, this one you can do L1, you can do L2. It's more just you're going to try to contain as much as you can, and then ones beyond that you're going to fit some sort of loss to. It's sort of an epsilon insensitive loss where above epsilon it's going to be this. So in that case, you would have to think about what sort of loss you want outside of the tube, outside of the regressor sort of domain where it's performing well. And if you have some estimate of how many that is, then that's a good number to put here.
That epsilon, is that as you drew it, the width of the tube and not the vertical distance?
You're correct. Sorry. It should be the vertical distance. So it's the width of the tube in the regression output. Sorry. Yes. All right. I'll quickly write down we'll also put a little bit, there's a W here. Well, we'll put a little bit of regularization on the classifier itself so it doesn't just to control that a little bit. It's often set pretty low. the way it's usually written, I'm going to use the notation, which is not as clear from this, but all the pieces fall into place, and it's how most of the papers write it. We're going to minimize this. So this is our regularization term. There's going to be some W that goes into the regressor, plus C, which is the balance constant between these constraints, our loss, and the regularization. So C higher means pay more attention to the data, and C lower means pay more attention to the regularization. New epsilon plus the average of, and there it gets a little bit strange. Each of the variables we're going to put I can never write this. This chi variable for the ones that violate. Each data point is going to have this attached. It's basically how much does it go outside the tube. Most of those will be 0. For anything in here, it's going to be 0 because we're minimizing this. And so sum of I to n. So sort of the average L1. So this is the L1 error. This is our fraction that are allowed to be outside. C balances regularization and classification, and this is the regularization term. Okay. I just wanted to put this up because if you go and look up support vector regression, you'll see something like this, and to see where the parts are mapping. If we square this, then it becomes an R squared loss, or an L2 loss function. Okay. Again, when we solve this, we put it through a similar machine as we used to solve the SVM. We're going to get something where only the points that are on the boundary and outside of the tube are used to constrain the solution. And W, again, is going to look like our regression output, or our regression projection direction, some set of alphas and the original points. Okay, so again, and these alphas will only be, again, non-zero for things that are outside the tube. So again, we've got a set of support vectors. In this case, we've got explicit control over how many there are. And just like before, we only need to use dot products. Same thing for the solution here. We only ever needed to use dot products. And so that's where now we finally get to the kernel trick. Oh, and I wanted to also introduce this idea of the slack sort of outside the tube variables. You can often, I wanted to show this, although it's a little bit confusing or a little bit complicated, because it'll happen here as well. In support vector machines, you'll often have slack variables for ones that are not, in the case where it's not linear separable, where you want to balance some regression and some fit to the data. Or sorry, regularization and fit to the data. So they'll have slack variables here as well, where things are allowed to violate the margin. Okay. We're finally to the trick. So all we used here were dot products in constructing the problems and solving the problems and then applying the solutions. So let's again set up a not separable problem as a classification problem. Yep.
Does the literature discuss like a deeper intuition as to why dot products are all that are necessary for these kinds of classifiers? I mean, part of it is it's sort of invariant to isometry. You can rotate the problem, make it sort of expand it out, bring it. You're interested in relative geometry between the data, not. That's sort of what it feels like to me. But
well, and we'll get to exactly that we can phrase this exactly in terms of relative geometry. I don't know if there's an intuitive explanation as to why. I don't remember one. And a lot of the papers that you'll read on this will have a bunch of stuff about non-intuitive aspects of it, like KKT conditions and things like this, about just making it solvable. But no, I don't know of one That is the intuition, though, that we're building up, that if we just use dot products, then we can use dot products to talk about similarity, and then we can just use measures of similarity to solve problems like this, based on similarity rather than actual data. That's where we're going to get to. Sorry, I should have probably introduced the trick before. But, you know, it's building up to the big finale. All right, so we've got some nonlinear problem here. I want to separate these two classes. It's called the axes A and B. We can't do this linearly, but if only we had sort of the distance from the center. And so let's change our data to, let's say that instead of taking the dot product of, you know, xi dot xj, let's use this. And let's call that a kernel function. Right? So I'm just taking the, sorry, it's a bit small. Taking the dot product of the points, I'm adding one, and then I'm going to square it. Okay. If we expand this out with A and B for 2 points, so we have, we'll A0, B0, dot, A1, B1, plus 1 squared. We can go through all the steps here, but in the interest of time, I'll just write out that this is the same as A0 squared, B0 squared, A0, B0, A0, this vector, where I've got some quadratic terms. Is that right? Yeah. Oh, there should be a comma here. Where I've got some quadratic terms and some linear terms in the one, and then dotted with the same thing, but now with ones. So it's still a dot product in a space where I've just taken this 2D points and I've turned them into 60 points. The quadratic terms, the mixed term, the linear terms, and the bias. Okay, so it's still a dot product. And I could do that explicitly for this problem. But I could also just, I did this just by taking the dot product and adding 1 and calling that something else. But I just showed that this, by expanding it out, is the same as a dot product in some space, this 60 space in this case. So why don't I just take this function, which I defined here, call it a dot product, and put it in my algorithm over here. Right? I say, I'm not going to use linear dot products, I'm going to use this squared, and this bias and square dot product. All of my algorithms only care about this function, right? By default, it's defined as just the linear dot product in everything we did here. But there's no reason, given the machinery, not to use this function or some other function. And that's the kernel trick. You show that your algorithms for the classifier or the regressor or clustering, PCA, only need dot products. Then replace the dot product with some other function called a kernel. There are certain constraints on what that kernel can be for these to still work. And really the only constraint is in some space, which may actually be infinite or extremely complicated, as long as the dot product is as long as it is a dot product somewhere, then all of this will just work. And you'll still get things like sparsity of the boundary and error tolerance and all of that. Excuse me. All right. So now that we know that, what can we do with it? There are, for the, I guess, 90s in this area, a lot of the work was, what are the interesting kernels that we can come up with? How can we apply them? How can we apply them efficiently? So let's let's show some of them.
Maybe before you do that, do you think you can just spell out why that particular function is such a great idea for that?
Oh, yeah, sorry. That's right. So once we have, so this particular function, right, where I add 1 and square the product, now I have these two terms, right? A0 squared and B0 squared plus the linear term, so I can compute exactly the distance from here. So if I take this and put it into the classifier, now I get a classifier that's basically any ellipse that I can build from these, right? I have all the pieces I need to make an ellipse, and I'm going to choose some W that weights each of these components individually. And from that, and then I'm going to bias it. And so I get a quadratic function in 2D space, and then I cut it off at some bias point. Sorry. So at that point, these become easy to separate. Is that clear? And it's just from the definition of just expansion of this dot product. This is called the polynomial kernel with order 2. It's a pretty common choice. All right. Thank you. All right. So what are interesting kernels? We've already talked about linear, and it is still useful. So there's just the linear kernel. Assuming your data is linear, it's always a good thing to check first. There are some nice things about linear kernels in that they can be computed much more efficiently than most others through some assumptions about the space. It's polynomial. Where you, through expansions of powers or whatever, you get the same sort of benefits there. If you think it's cubic or something, this is also a good choice to experiment with early because maybe something simple. For people doing genomics, the string kernel. So I'm going to take all possible k-mers of length n, and I want to look at two strings and compare and see how similar they are. I'm going to count that each k-mer. And see how much it occurs in one versus the first string, the other string, and take their product, and then sum that up. This is the dot product over basically a vector of all the counts of every k-mer, but you don't need to compute that vector. You just compute the counts, iterating over the two strings. Radial basis functions are a very common choice for SVMs. This is based entirely on sort of a local density estimate of the points. So you basically put around each point a little Gaussian blob with sigma, and then project into a space where similarity is defined by which points you are close to and which points you are far under that sort of Gaussian blob. So this point is close to this point, but far from this point. That can give you some very interesting sort of wavy boundaries based on sort of projection onto this basis function. This is very common. This is one of the most common sort of multidimensional basis functions to use. If you're working with graphs, the heat kernel, which is some t that you have to set, much like sigma, and this is just the Laplacian of the graph, or the graph Laplacian. So based on connectivity and labels on graph nodes, you can infer other graph nodes based on how close they are to each other, and then it's sort of the graph-based equivalent of or similar to the graph of the RBF. But really, you can just search for kernelized anything. You'll get a paper from the 90s or early 2000s, or maybe more recent, and it probably has something like kernels for comparing graphs, kernels for, SVMs for such and such. There's a lot of literature out there. They're fairly powerful. They're good baseline, excellent baseline methods, and often the best performing in a lot of cases. I'm not going to go into I was going to talk about approximation, because one of the problems is when you do this, what I swept under the rug is we only need dot products, but to solve these in the default form, or the sort of naive ways, you have to build the full kernel matrix, it's called, every pairwise dot product, which is fine if you have 20 or 100 or 1,000 examples, but gets a little hairy if you've got 10,000 or 100,000 or millions of examples. And so what you usually do is subsample some set from your data, just ran uniformly or by other methods. And instead of building that full kernel matrix, you build sort of a condensed low-rank approximation. That's called the Nystrom method. And it makes it much more efficient. And in fact, if you sample few enough, you can use any kernel, use a Nystrom approximation, project into that very sparse, that very low rank set of space, and then just apply the linear kernel. And it's the same as if it's approximately the same as if you used any other more complicated kernel, but for a much smaller projection. The Wikipedia page, I think this is pretty good, and there are a bunch of lecture notes out there as well. Which part? Oh, so
So you claim that you have a commutative diagram that's approximate. What is that diagram?
That diagram is this, the kernel matrix, so all pairs of all, right, looks like, actually there's a nice diagram that I should just copy out. All right, all pairs of all data points under the kernel. Subsample a set of columns. Okay. And it's symmetric, right? It's dot products. It has to be a dot product, so for this to all be valid, so it's symmetric. So I'm at the same time sampling this set of rows. Okay. This kernel is approximately the same as, so I'm just taking these parts and stacking them there. And this is all the data, right? So it's this column and this column, or these columns stacked, times the core. I think this is a pseudo-inverse of just this block, this block, this block, this block, okay? And then the transpose. So the rows. Okay. You can construct this implicitly and then put it in your algorithm, so you subsample this and do all your work and just use this representation to solve completely. Or you can actually, if you subsample small enough, it may just be worth using that as a new feature space. So the similarity of some number of landmark points to your other points, and then put it into a linear kernel after some normalization based on this. This is how, there's an SVM example in sklearn, and it works like this. It basically applies a linear approximation, or sorry, it applies a linear kernel to this approximation after some normalization. Okay. Other questions before I wrap up?
That works with any random subsample of columns?
It works with any random. There are various guarantees. You can do better. There are various papers on how to select those columns, or how to select multiple subsets of columns and then compute how good the approximation is and merge them together based on that. but just random subsample works.
Yes, so the trick that you said about adding one and then inputting that as instead of the dot matrix. So have you compared how were the changes in terms of performance or accuracy or classification to be able to use this instead of the existing kernel system?
So this will be strictly more accurate and therefore more prone to overfitting, right? Because I can still do the linear product here. So at that point, it becomes a tuning problem. There's still, it doesn't remove the need to tune to prevent overfitting or to do some regularization or things like this. So you still have to do cross-validation and try different values and things like this. Often the defaults work pretty well in a lot of it, so.
And also, like with the SVM, there's a big question about dimensionality. So in genomics data, especially patients data, you have very few samples, and your dimensions are very high as your genes or your SNPs or whatever you are using. So what kind of kernel would generally, like they say you can go for linear, or RBF tends to overfit, or even doesn't do well as well. So what's your suggestions about how do you kind of figure out that problem using this trick or something?
I don't think I can answer that. I would search the literature, see what other people have done. Yeah, I would have to look more deeply at it. If people haven't applied it, then I could maybe extend other knowledge into that, but I'm not familiar enough with the problem.
Okay, just in the interest of time, we're gonna have to move on. If you have other questions, though, maybe catch Ray during the food break, something like that. So thank you very much, Ray.
So thanks, Akil, for the introduction, and thanks for having me here. It's great to be here, and thanks, Ray, for the great primer. So as Akil said, right now I'm at Harvard, but I'm actually going to speak about work that I did in my previous life at Eran Sega's lab at the Weizmann Institute of Science in Israel. And this work was done with Daphna Rothschild and Elad Barkan, the other two co-authors of the paper, which is about to be published in Nature today, actually, at 1 p.m. So this is a very timely talk, hot off the press. Yeah, and so this talk is about linking gut microbiomes, genomes, and phenotypes via linear mixed models and kernel methods. All right. Yeah. So this talk is about the gut microbiome, and I assume that most of you heard about the gut microbiome. So very briefly, so the gut microbiome is a community of mostly bacteria that lives in our guts, mostly in the colon, and they are intimately associated with our health. And in recent years, it has become very important. And how can we know that? By the number of covers on which the gut microbiome featured. So it featured on Science and Nature, and the magazines like New Yorker, The Economist, Scientific American, and Nature again. And it has been called our other genome. And to make this claim that the gut microbiome is important, more scientific. So here's a graph I pulled off Google Trends a few days ago. So apparently the heydays of the human genome were back in 2004. And now it's the time of the gut microbiome. But it looks like CRISPR is about to overtake us all soon. Yeah.
What's the periodicity?
What's that?
What's the periodicity?
What do you mean?
Summer.
What do you mean?
Like, why does it go up and down?
I have no idea. This is just, I put it off Google Trends. Yeah, I wouldn't trust this too much, but it's interesting to look at. Yeah, so more seriously, the gut microbiome is important because in recent years, we've learned that it's really intimately connected with many aspects of our health and with many diseases, diseases like diabetes, obesity, non-alcoholic fatty liver disease, inflammatory diseases, metabolic diseases, aging, and even stuff like cancer and neurodegenerative disorders. And the gut microbiome also made a lot of headlines in recent years because it turned out that it's a very, very effective treatment for C. difficile infection, which is a very dangerous infection in the gut, and the gut microbiome has over 90% effectiveness when doing fecal microbiota transplantation, which is a very hot topic right now. So I hope that I convince you that the gut microbiome is a big deal right now. But even though it's a big deal, we still don't really understand many aspects of the gut microbiome. And the reason is that it's complicated. So if we think about the main aspects and factors that are involved with our health, so we have our genetics and our age, but then we also have our lifestyle and diet and our phenotypes and the gut microbiome. And there are many rich and complex interactions between all these factors. So we know that our genetics and our age have a causal influence on these factors at the bottom. But then there are also these complex bidirectional interactions between these factors at the bottom and each other. So it's pretty difficult teasing apart what really is associated with what. And this is basically what we try to do in this work. And more concretely, so this work is roughly composed of 4 different parts. The first part is about studying host genetics microbiome associations. So this is the classic question of nature versus nurture applied to the gut microbiome. So how much does the gut microbiome, how much is it shaped by our genetics? And the second question will try to quantify the association between the gut microbiome and our lifestyle and our diet and so on. And these 2 parts will form the first half of this talk. And in the second half, we'll also bring phenotypes into the game, and then we'll try to quantify the association between the gut microbiome and our phenotypes. We'll try to come up with the gut microbiome analog of genetic heritability. And in the 4th part, we'll kind of tie it all together by trying to predict phenotypes using all of these factors and seeing how much the gut microbiome contributes over all these other factors. These are the 4 parts of this talk. And just to put things in context, so this project is part of a large ongoing project at the Weizmann Institute of Science in Israel called the Personalized Nutrition Project. And this project aims to understand how our diet affects our health. The main emphasis is on glucose measurements because we know that high glucose levels are the main determinant of diabetes, which is a worldwide healthcare problem. And this project requires a lot of participation from its participants because every participant needs to provide blood samples and stool samples to profile the gut microbiome and to fill very detailed questionnaires. But on top of that, they have to be connected to a continuous glucometer that continuously measures the blood glucose levels for at least 1 week. And during that week, they have to log every meal that they have on an app on their smartphone, and this allows really associating what they eat with their glucose levels and other aspects of their health. So it requires a lot of participation on behalf of the participants. So up until now, about 1,000 participants have been recruited, only healthy individuals, so individuals without diabetes or other kinds of diseases related to the gut microbiome. And over 50,000 meals have been logged and over 2 million glucose measurements, and the project is ongoing. And what we did is a specific study, which is a part of this large project. And more specifically, let's look at the data that we had in our study. So we had about 1,000 participants, and for every participant, we had profiles of their gut microbiome collected using 2 different techniques. So both 16S ribosomal RNA gene sequencing, which is a pretty inexpensive technique, but has lower resolution. But we also had metagenomic sequencing of the gut microbiomes. So this is essentially shotgun sequencing applied to the gut microbiome. So we essentially had the entire sequence of the gut microbiome of every participant, which is cool. And these participants also had to fill very detailed questionnaires about dietary habits and lifestyle and medical history and so on. And we also genotyped all the individuals at about 700,000 SNPs, and then we imputed these. And they provided blood samples, which we used to measure stuff like fasting glucose and lipids and HbA1c, which is a measure for diabetes risk, and body measurements like weight and BMI and waist hip circumference. And this is the data that we had. So we have a lot of data. And in microbiome terms, this is one of the largest cohorts in the world right now, the first or the second, depending on how the two cohorts increase in size. And because this is an MIA talk, so I'm going to say a few words about how our data looks like when we represent it on the computer. So genotypes are relatively easy to represent because we have a bunch of loci, and at every locus we have 2 alleles, and we can just represent it as 2 strings or as a vector, like people have been doing in GWAS for over a decade now, and it's pretty straightforward. But representing the gut microbiome is a bit trickier because you may remember from Biology 101 that we can classify bacteria according to taxonomic ranks. And so the usual way to represent gut microbiomes is using relative abundance of bacteria because the absolute number of reads can be very different between different samples for technical reasons. So what we do is normalize all these reads. So what we care about is only how abundant is one type of bacteria related to another type. So we don't care about the absolute counts, only the relative counts. So it's called relative abundance. But we can represent these relative abundances at different taxonomic levels. So the top taxonomic level is typically the phylum level. And in this project, after filtering, we had about 7 different taxa at this level, meaning that we can represent the gut microbiome of every individual using 7 different numbers. But we can go down to the lower taxonomic levels, and then we can get, for example, to the genus level, which has about 100 different taxa after filtering. And below that is the species level, which has about 300 different taxa. And because we had metagenomic sequencing, we can drill down even further and profile the relative abundance of specific bacterial genes. And then we have millions of numbers, millions of variables, which can be used to represent the gut microbiome. And so we have this trade-off because if we choose to represent individuals using the upper taxonomic levels, this is a pretty robust and low-dimensional representation that is easy to work with, but it has a pretty low resolution. But if we go down, then the resolution becomes much higher, but it's probably a bit noisier, and it's much more difficult to work with because it's a very high-dimensional data. And so, and this decision will form a large part of this talk, mainly of the second part of this talk. Yeah?
Is there any collapsing you can do across like orthologous genes at the bottom level that would make that interesting, to make that number smaller, but collapse across different bacterial species?
Yeah, you can probably do that, and we did something somewhat similar, but this will show up mostly at the second half of this talk. So let's get back to that in like half an hour. All right, but in the first half of this talk, actually, we chose to use the species level because this gave us a nice trade-off between high resolution and representation that is relatively manageable to work with. All right, so all of that was the introduction, and so let's step into the results. And as I said, the first part of this talk is the classic question of nature versus nurture as applied to the gut microbiome. So we want to ask how much is the gut microbiome shaped by our genetics? And this question actually has very important clinical implications because of fecal microbiome transplants, which I told you about. So right now, we don't really know if the genetic compatibility between the donor and the host is an important factor. So here is a spoiler alert, no. But let's look at the results. But before looking at the results, I will just mention, so this entire study was done in Israel, and Israel is an interesting testbed for these kinds of nature versus nurture questions. And the reason is that the population of Israel is pretty genetically diverse, and that's because most of the Jewish population arrived over the last 100 years. So broadly speaking, there are 5 major ancestries, Ashkenazi, which arrived from Europe, and Sephardi, which arrived from mostly Turkey and Greece, and surrounding countries, and North Africans from here, and Middle Eastern from here, and Yemenites, which arrived from Yemen here. And there are some other smaller ancestries, but these are the 5 biggest ones. And so we have a lot of genetic diversity because these ancestries were kind of separated for at least hundreds of years. But on the other hand, the population is relatively environmentally homogeneous. And that's because of the cultural history of Israel. So people from different ancestries, for the most part, live more or less, they lead the same lifestyle, and they eat more or less the same kinds of food. So we don't really have a reason to believe that ancestry is associated with environment. And this is cool because this means that this population, in some sense, approximates the ideal experiment that we would want to conduct. Because the ideal experiment would take genetically diverse individuals and place them under exactly the same environmental conditions for a long time. And this would really allow us to tease apart the effect of genetics and of environment. So we can't place humans in petri dishes, but this population is a kind of an approximation for doing that. All right, so after preparing and cleaning and pre-processing the data, so we started analyzing it, and we took the genotypes and we computed the top principal components. And what you can see here is that we got a very nice correspondence between the genotypes and the self-reported ancestry. So you can see that we had a large cluster of Ashkenazi individuals here, and here we have individuals from North Africa and from Middle Eastern individuals and Yemenites, and here we have Sephardis, and most of these other individuals are admixed individuals, so they're just some kind of mixture of these ancestries. So far, it all looks great. So the ancestries are strongly reflected in the genotypes, and then we tried to do the same thing for the gut microbiomes, and we didn't see anything. So there was no correspondence between these top principal components and the gut microbiome composition, meaning that individuals from the same ancestry do not have more similar gut microbiomes to each other than just a random pair of individuals. So this didn't look too good. So we decided to just look at the ancestry, to not even look at the genetics. For every individual, we had the country of birth of each of their grandparents. And so we could compute the fraction of shared ancestry between every pair of individuals according to the number of grandparents from the same ancestry. And so what you can see here are box plots where we took pairs of individuals, all possible pairs of individuals, and we computed the microbiome dissimilarity between them, which is a popular measure in microbiome and in statistical ecology. This is a Bray-Curtis measure. I'm not going to get into the details. This is a popular measure of microbiome dissimilarity. And these are box plots over all pairs of individuals, but as you can see, there's no signal here, right? So individuals from the same ancestry do not have more similar gut microbiomes. So this also didn't look good. And so we forgot about the ancestry, and we just computed the genetic kinship between every pair of individuals. So what you can see here, so every circle is a pair of individuals, and on the x-axis, we can see genetic kinship, which is a common measure of genetic similarity. And on the y-axis, again, we have microbiome dissimilarity, and again, there is absolutely no correlation. Right. So at this point, we did what I guess every scientist does at this stage, and we freaked out because this didn't look good, and this also seemed to contradict the literature. And then began a very lengthy and annoying stage of going back to the data and trying to look for bugs in our data or in our analysis or somewhere. And we were really concerned about sample mix-up. But we had a secret weapon that most GWASs do not have, which is that every stool sample is contaminated with some human DNA. And so we could use that to extract the human DNA from the stool samples and then match the human DNA from the stool samples with the human DNA from the blood samples, which we used for genotyping. And this was a very lengthy process for all kinds of reasons, but we did that, but there was no mix-up eventually. And then we also tried to associate the gut microbiome with the phenotypes and with environmental conditions, just a second. And again, we could see a perfect match, and this actually formed the beginning of the second half of this talk. So eventually it was nice, but at this point we were freaked out. Yeah.
So yeah, that's a very interesting graph and disappointing as well. But what is the age group are you looking at here? What is?
Yeah, so I think it's between 20-something, 24 and 60, if I remember correctly. Yeah.
As you know, like as you already know, the microbiome changes over time consecutively. If this study was done in infants, for instance, 1,000 infants, would you expect the microbiome to be associated with the parents or stuff like that, so that a link between genetic?
Yeah. So that's a good question. So there are many confounders, right? Because the gut microbiome is associated with many factors. That's what this talk is about. Age and the diet and many other things. And we tried accounting for these confounders in many different ways. And I'm giving you the short version of all of this because we did much more analysis. But no matter how we cut and slice the data, we didn't really see an association with genetics or with ancestry. And all right, so we were really concerned because there was a series of really beautiful papers which showed that monozygotic twins share much more gut bacteria than dizygotic twins. In the sense that there are some gut bacteria that are shared much more across monozygotic than across dizygotic twins. And we know that twin studies that compare monozygotic to dizygotic twins are the best, the gold standard for estimating genetic heritability. So we had no reason to doubt these papers. And so what we did is, so we suspected that maybe even though there are some heritable gut bacteria, they form only a small minority of the overall gut microbiome. And so we asked Julie Goodrich, the first author of these two papers, to get us access to the data. And then we took this data and we tried to come up with some rough measure of the total microbiome heritability. So what we did is we devised this measure for the total microbiome heritability, where we iterate over all the bacterial taxa over that we consider as being heritable according to some criterion. And for every taxon, we just multiply the relative abundance with the estimated heritability based on the results reported in the previous paper. So we didn't do the analysis ourselves to not jinx it. And so, all right, so what we did is, so we took all the bacterial taxa from the Twins UK dataset, which is a dataset of around 1,100 twin pairs from the UK, and we sorted them from most heritable to least heritable. And what you can see here is, so on the x-axis we have heritability p-value cutoff, which means that at every point we consider only the taxa with heritability p-value smaller than this cutoff. And on the y-axis we can see the cumulative distribution of two curves. So the dark curve is the microbiome abundance. So by definition, when we use all the taxa, we account for 100% of the abundance by definition. And the light curve is the estimated total heritability according to this very rough measure. And what you can see here is that basically no matter where we cut it, the total microbiome heritability according to this measure is not that high. It's smaller than 20%. And for comparison, the estimated heritability of height from twin studies is 80%. Yeah.
What's the jump in the blue curve around somewhere between 10 to the -2 and 10 to the
That's a good question, but I don't know. Actually, I don't remember. Some very abundant taxon. But we can check it. I don't remember. Anyway, so choose your cutoff, and then you will get some estimate. So one reasonable cutoff is the 5% FDR cutoff. And at this level, the total heritability is 1.9%. And so this was our lower bound. And then we also devised an upper bound, which is just a p-value of 0.05 with no multiple testing correction whatsoever, which is a very liberal cutoff. And then we get to 8.1%. So the truth is probably somewhere, here in the middle. We don't really know where it is, but when we saw this, we became much more relaxed, because this means that apparently, even though some gut bacteria taxa are heritable, or clearly heritable, they form a pretty small minority of the overall gut microbiome. Yeah?
But did the heritable taxa show ancestry stratification?
No, they didn't in our data. But actually, the taxon that was reported to be the most heritable, and it accounted for a large fraction of this signal. It's called Christensenella. And for technical reasons, metagenomic sequencing does not have access to Christensenella. And that's because of very technical reasons, because metagenomic sequencing can only profile. There are some taxa that cannot be profiled yet. So yeah, maybe we missed Christensenella, but other than that, we didn't really see a signal. But we didn't have twins, so we had, in some sense, less signal in our data than we would have had we have twins. Yeah.
What are you defining as a species using your 16S data set?
Yeah, so we could do that, but we had less individuals with 16S data than we had individuals with metagenomic sequencing data. We had about 60% of the individuals had 16S data, and we repeated all the analyses also in the second half of the paper with 16S data, and the results were reproducible. But actually, I mean, I'm not at all doubting that Christensenella and all these other taxa that were reported in the literature are clearly heritable. What I'm saying is that probably these form a small minority of the overall gut microbiome. So and we try to take a more global approach and to try to see if the microbiome is heritable as a whole. And the reason is, I mean, I think it's more interesting, but there are also lots of statistical considerations here, which I don't have time to go into. But if we do many different analyses, then we have a severe multiple testing correction that we need to apply, and our data set is it's large in microbiome standards, but it's not that large in GWAS standards. And so we will lose power if we just try testing all the hypotheses out there. And that was our these were our considerations, because the fact that some bacterial taxa are clearly heritable, that's already established, and we don't doubt that. What we try to do is to take a more global approach. All right. And so we did actually many more tests that I don't have time to go into, because I also don't think that it's very interesting, because we did try to look at associations with specific taxa and with specific SNPs, and we didn't really see an association. But I'm not saying that it doesn't exist. All I'm saying is that these associations are probably weak and difficult to identify, meaning that donor-host compatibility is probably not very important, unless maybe, you know, Christensenella, which is very heritable, is the bacteria that is the one that is most important for the honor-host compatibility. We don't know. But as a global entity, we decided that we reached the conclusion that the gut microbiome is not strongly shaped by our genetics. But if it's not shaped by our genetics, then it should be shaped by the environment, right? So we wanted to try to see that to get some positive control for all of this. So this is the second part of this project, where we try to associate the gut microbiome with lifestyle and with diet. And we could do that using two different approaches. So first, we had a few dozen individuals in our data who reported living in the same household during the study. And we also had a few dozen individuals who were first-degree relatives, and we also actually excluded all these individuals from most of the analyses, but here we actually used them directly. And we also had about 100 or 200 individuals who were second to fifth degree relatives. And so what we did is, again, so we took all the pairs of individuals and we stratified them into either first degree relatives or second to fifth degree relatives, or pairs sharing a household, or all other pairs. And again, on the y-axis, we have microbiome dissimilarity. And what you can see here is that individuals who are either first-degree relatives or who share a household have much more similar microbiomes to each other compared to the typical pair of individuals at the species level and at the bacterial genes level, but actually not at higher levels. Yeah.
Did you do the same genetic analysis to see whether the first-degree relatives actually might be more similar for genetic reasons?
That's a good question. I'm not sure that we did. But I mean, by definition, we computed first-degree relatives by computing the genetic similarity. So actually, yeah, by definition, we did that, sorry. By definition, we computed the genetic similarity between every pair of individuals, and that's how we could stratify them into first-degree relatives, second to fifth-degree relatives, and so on. And so we made sure that all the individuals with shared household are not also related. If they were related, we didn't include them in this analysis. So yeah, we did that. Yeah, so the individuals sharing household are, by definition, not related, even to a fifth-degree relationship. Yeah.
Why is the microbiome dissimilarity so much smaller at the level?
Yeah, that's a good question, but that's because of technical reasons. So we know that at the species level, we have around 300 taxa. At the bacterial genes level, we have around 1 million genes, or even more, depending on how we apply the filtering. So these are very different objects, and they give pretty different results because of pre-processing. Yeah, all right. So finally, we got some positive results. So that was nice. So this clearly shows that individuals who are either living right now in the same household or lived in the past in the same household are, have more similar gut microbiomes compared to the typical pair of individuals, which we think is a pretty solid evidence for environmental effects. And then, because these individuals filled all these very detailed questionnaires, so we finally put them to good use. And so in the next analysis, we just took a lot of environmental variables that we had mostly from the questionnaires, but also from other sources, like blood tests. And we tried to explain the variability of the microbiome using a method called Permanova, which I don't really have time to go into. And we could explain over 20% of the variability of the gut microbiome using all these measures. And that was very statistically significant, I think 0.001. And then we tried to replicate the same analysis with genetics, and we couldn't get to any statistically significant result. And what you can see here is the variance that could be explained by specific categories of measurements. But we need to be very careful about the interpretation of this, because you can see that different categories have different numbers of measurements inside them. So this is obviously a very strong confounder. But we think that the more interesting result is that we could explain the gut microbiome variability using environmental factors, which just goes to show that the environment is a much stronger determinant, apparently, over the gut microbiome than host genetics. All right.
What about the reverse causal direction?
Yeah, that's a very good question. Yeah, I should point out the very important caveat that in this project, we're not really speaking about causality. I mean, for genetics, we do, because I can't imagine the gut microbiome changing our genetics. But for everything else, we're clearly not trying to indicate causality. So we're just quantifying associations. For all we know, maybe our environment shapes our gut microbiomes. So let me wrap up this part, the first half of this talk. So we reached the conclusion that the microbiome is mostly shaped by environmental factors, and the total microbiome heritability, depending on how we define it, is probably around 2%, which is in some sense bad news for us, but it's very good news for medicine, because this probably means that microbiome transplants can be carried out without worrying too much about donor-host genetic compatibility. And our hypothesis is that the microbiome genetic architecture is polygenic, meaning that the effects are spread across many different variants in the genome with relatively weak effects, because most complex traits have a polygenic architecture, and the microbiome is pretty complicated. So that's our hypothesis, but we need much, much larger samples than we have right now to verify this. All right, and now we're switching gears, and we're moving into the second half of this talk, where we're also bringing phenotypes into the game. Yeah.
I'm not sure if you mentioned this already, but did you compare, like, just like based on what age your samples were from? Was there more heritability in infants than in the older people? Like, I would imagine the longer, is that something you could even test easily, or?
the main problem with doing all these kinds of tests is that we have a relatively small sample in GWAS terms, 1,000 individuals. It's huge in microbiome terms, but it's very small in GWAS terms. So if we cut and slice our data in many different ways, then we lose lots of statistical power. So we try to be careful, not to test million hypotheses and then not being sure if our findings are true or not. So maybe it is, but we need larger samples to really be sure. All right, so now we're bringing phenotypes into the game, and I personally think that this part is more interesting. So what we try to do in this part is to come up with the microbiome analog of genetic heritability. So what we wanted to do is to quantify microbiome phenotype associations after accounting for lots of other factors. So here is a rough illustration of what we want to do. So we're going back to this illustration, and then we want to explain the portion of the phenotype that is explained by genetics, and then we want to explain the portion that's explained by age, and then the portion that's explained by diet, where we approximate the diet via the median daily caloric consumption of different types of food. And then we want to ask how much of the remaining variance can be explained with the gut microbiome. So this is what we want to do in a nutshell, and it's by design supposed to be very similar to genetic heritability. In order to do that, so I'm going to give a very brief review about how we typically estimate genetic heritability in GWAS. So what we have is we have a vector of phenotypes, one phenotype for every individual, and we assume typically that this vector follows a multivariate normal distribution, which is the generalization of the normal distribution that you all know of probably. So instead of a mean, we had a vector of means, one mean for every individual, and typically this vector is affected by risk factors like, for example, age or diet or smoking status. And instead of a variance, we have a covariance matrix. And typically, this matrix is given by the sum of two matrices. The first is a matrix of genetic covariance, and the second is a matrix of environmental covariance. And the idea behind this model is actually very simple if you don't want to get into the math. The idea is that we assume that individuals who are more genetically similar are also expected to have more similar phenotypes. That's the entire idea in a nutshell. And so this matrix of the genetic covariance essentially is just a fancy way to measure genetic similarity. And then given all of this, we define heritability as the magnitude of the genetic covariance matrix divided by the magnitude of the sum of these two matrices. So this is the idea of genetic heritability. And now we wanted to do basically the same thing for microbiome. So again, we have this vector of phenotypes, which is normally distributed, but yeah.
Just to clarify one thing. In this picture, you haven't drawn the parameters that we're actually fitting, but my understanding is we have a coefficient on the genetic similarity matrix, a coefficient on the environmental similarity matrix, and we're just asking what proportion, once we fit those two coefficients, what proportion of them is on the first?
That's absolutely correct.
Okay.
Yeah. And now we want to do the almost the same thing for the microbiome. So again, we have this normal distribution, but now our mean vector is given by the sum of two vectors. So the first, like before, is a vector of risk factors like diet and age. And the second is a vector of polygenic risk scores, which is how we encode the genetic effect on the phenotype. And so for every individual, we have a polygenic risk score, which I will speak about in a minute. And now the covariance matrix is given by the sum of a bacterial covariance matrix and an environmental covariance, which I forgot to say before, but this matrix is essentially supposed to capture everything that we couldn't capture with the other matrix. So it's all the noise, all the things that we can't explain go in here, and this is a diagonal matrix. And exactly like before, the underlying idea is that individuals who have more similar microbiomes are expected to have more similar phenotypes. So it's a very similar idea. And I will just mention that these polygenic risk scores were computed using available summary statistics from previous GWASs based on tens of thousands of individuals, which is how we overcame the relatively small size of our sample compared to other GWASs. So they gave us pretty accurate estimates compared to anything that we could have done using only our own data. Yeah.
Do you have any sense from like literature or from analyses you've done how much of this is causal in the different directions, or any like intuition for?
That's a good question, and this is very hotly debated. So the short answer is no, we don't. We don't have any idea. But there are some studies that show that by perturbing your microbiome, you affect your health. So there are obviously some interactions, causal interactions, but really quantifying them, I mean, as far as we know, we are the first study to really try in a principled manner to quantify this association whatsoever. So obviously no one did it with directionality. Yeah. And as before, we define the microbiome association index as the magnitude of the bacterial covariance divided by the magnitude of the overall covariance. Yeah.
So you're including polygenic risk as a fixed effect. How similar is that to just looking at the residual phenotype?
It's probably, what you're suggesting, I think, is an approximation for this, because here we fit everything jointly, and then we do a two-stage process, which is less accurate. Yeah. Yeah.
You have this, you're in this rare situation where you actually know something about environmental covariance.
Right, right. So everything, that's a very good question. So that's an excellent question. So we have risk factors, and I didn't say what these risk factors are. I'm saying risk factors, but these risk factors mostly capture environmental effects, and we mostly accounted for diet, because we know that diet is a very major determinant for the gut microbiome. And I said that briefly, but what we did is, so we mostly included the median daily caloric consumption of different types of food, which, based on the literature, is a very good proxy for diet. And I'm saying environmental covariance, but this is very similar to the epsilon that you have in linear regression. You have to, you must have some noise term that just captures everything that wasn't explained with all the other stuff, right? But we do have age and gender and diet encoded here. So these are the...
You chose to include sort of environmental similarity as fixed effects rather than random effects.
Yes, yes, we did. Yeah, yeah, exactly. All right. So we know exactly how to compute microbiome association index now, instead of, except for one thing, that we don't really know how to define this matrix. So in genetic heritability, it's pretty well established by now, but we are the first to do this for the gut microbiome, so we had to make some decisions. And it's not at all clear or straightforward how to do this. So this means that now we need to decide how to encode bacterial similarity between individuals. And so I'm going to mention as an aside that measuring bacterial similarity, or actually dissimilarity, is a very common task that's done in statistical ecology. And there is this term called beta diversity, which for the purposes of this talk can simply be defined as a measure of bacterial dissimilarity between individuals, although this term goes back decades, and it has much broader implications than what I'm speaking about here. But for our purposes, it will just be bacterial dissimilarity. And there are many different metrics that people use. And this field goes back decades, way before genetics, actually. And probably the most common one is Bray-Curtis dissimilarity, which is defined like this. So for two vectors, U and V, this is the distance measure. I'm not going to get into the details, but essentially, it's an L1 measure, but that only takes into account the bacteria that are actually present in one of the two individuals. So the reason I'm showing this is to show that measuring bacterial microbial dissimilarity is a very old technique in statistical ecology. But we wanted to connect that with the method that we use to estimate genetic heritability, which is typically called linear mixed models, by the way. So we wanted to connect what people usually do in statistical ecology, which is these kinds of measures and analyses based on them, with what people usually do in GWAS, which is using linear mixed models exactly as I showed before. And so here I'm going to show how this connection is made, and this is where it also fits to Ray's talk from before about kernel regression. So we're going to use the law of cosines. This, by the way, is the most mathy slide in this talk, so bear with me. So the law of cosines says that the square of the Euclidean distance between two vectors u and v is given by what you see here. It's not interesting per se, but what is interesting is that if we assume that u and v contain random samples from two distributions of two random variables, x and y, then we could rewrite it like this. And this is cool because this allows us to connect the law of cosines with covariance matrices. So given a distances matrix D of Euclidean distances, then the covariance matrix should obey this equality, which is exactly what's written here. I just switch the sides, and this operator is an element-wise multiplication. So this is just the matrix D where we take every entry and multiply it by itself. And this is just another way to write this, and it gets a bit technical, but the equations aren't really important. What I want to say is that we have a very simple way to find, given a similarity matrix D, the corresponding covariance matrix. And all of this leads to the fact that what I just showed you is actually another way to describe the kernel trick, but using a distance metric instead of the kernel that people typically think about when they're speaking about the kernel trick. Because this is cool. This means that now you can take any distance metric that you want and to convert it into a covariance matrix and then plug it into the linear mix model that I showed you before. So this is really a nice way to bridge these two pretty separate fields together. Yeah?
Same math as in multidimensional scaling, where you start with similarity and then convert it into something that you can take the singular value decomposition of?
This is a good question. My rough answer is that not exactly. I think in very broad terms, but I think the mathematics is pretty different. Yeah, because I mean, one justification is that this is all done in closed form formulas. In multidimensional scaling requires some third space.
The linear case.
Yeah, the classical case is.
Oh, yeah. The linear case is pretty much equivalent to PCA, right? Yeah. So, yeah, this is very deeply connected. There are lots of very interesting connections, which I think I don't really have time to go into. I will mention that this is cool because this is related to one of the questions that someone asked Ray about using the kernel trick in high-dimensional spaces. And I'm going to show that the real magic of this kernel trick for our purposes when working with genetic data is that this is a very convenient way to work with high-dimensional data. I mean, kernel methods in machine learning have kind of fallen out of favor because of deep learning. But I think that here in genetics, they still really shine because of this consideration. So our conclusion is that all we need now is to come up with a good dissimilarity measure, possibly from one of the measures that people use in statistical ecology, and then we could plug it into the linear mix model. So we needed to come to decide what is a good similarity measure, and then we went back to this slide that I showed at the beginning because we had to make two decisions. One decision is what taxonomic level to use to measure similarity, and the second question is how to measure similarity. And because we are the first to do this, we had no idea what's going to work, so we tried many combinations, but we used out-of-sample likelihood to find the combination that seems to work best, meaning the combination that explains, the best explains phenotypes in order to not overfit. And actually, the measure that we came up with is not based on one of the measures used in statistical ecology. What we came up with is to take the level of bacterial genes, which means that we have millions of variables, and then we consider every gene is either present or absent if its relative abundance is above some cutoff. And then for every individual, we have this really high-dimensional binary vector, and then we use these binary vectors to compute Euclidean distance. And this is how we defined our covariance matrix. And so this means that statistically, this is cool because this is a very nice intractable way to include millions of different variables in our model, because all these millions of variables are just used to define the similarity matrix, and then it all fits into the linear mixed model framework. Yeah.
So in the dissimilarity matrix, right, you showed that people who shared same household, they had similar microbiomes. So before starting to calculate the dissimilarity, do you sort of correct for these kind of things?
Yeah, that's a good question. We excluded all the individuals sharing a household and all the first degree, actually all the up to third degree relatives from all these analyses to not confound things with household and with genetics.
But that is the only thing you know, but what are the other?
Yeah, but we included the other main determinants that we know of, which is diet and age and gender, as fixed effects, which is the question that John asked before. Yeah. Yeah.
Do you at any point factor for the abundance of the different bacterial species per individual?
So we use relative abundance here, right? So
Relative abundance across your samples.
For every individual, the vector sums to 1. Because the total number of reads is
and that factors towards your comparisons.
Yes.
Like if there are changes to the relative abundance.
Yes, yes, it does.
Okay.
Yeah. So statistically it's cool, but from a biological perspective, we also think that's cool because the fact that genes gave us by far the best results means that the functional composition of the microbiome is much more informative towards explaining our phenotypes than the taxonomic composition, meaning that for our purposes, I don't care what a bacteria is called as long as I know what genes it has. Meaning that for our purposes, a bacteria is just a collection of genes. We don't care about anything else, which is surprising and interesting. We didn't expect that. All right, so now you know everything we need to know about how to compute microbiome association index. And so we went ahead and estimated it for 12 phenotypes. So we have HDL cholesterol, lactose consumption, waist and hip circumference, glycemic status, BMI, waist-hip ratio, fasting glucose, HbA1c%, creatinine, height, and total cholesterol. And why did we choose these 12 phenotypes? Because we had available genetic summary statistics for them. We had many, many other phenotypes, but without genetic summary statistics, so we couldn't compute the polygenic risk scores, and so we couldn't tease apart genetics from gut microbiome effects. So you can see the estimates here, and you can see that for 8 out of these 12 phenotypes, we got pretty large and very statistically significant estimates. And here on the right, you can see the 95% confidence intervals. And we think it's cool. So we went and compared these estimates with genetic heritability estimate from the literature. And what you can see is that these are roughly comparable for many of these phenotypes, not for all of them, but for many of them. And we think that's a big deal, because what this means is that the gut microbiome statistically explains similar proportions of phenotypic variability as our own genetics, which we think is a big deal, but with the caveat that it explains statistically because we don't indicate causality. But we still think that's really, really interesting. I mean, people have been speaking about this and making this argument for years, but as far as we know, this is the first principled way to actually estimate and to quantify this association in a principled manner. All right. And now that we finally got some really nice and exciting positive results, so we wanted to replicate them. So we collaborated with a really great Dutch research team from University Medical Center Groningen, which has a similar cohort to our own called the Lifelines Deep cohort. It's actually the only cohort in the world that is similar to our own, in the sense that they have around 1,000 individuals with genotypes and metagenomic sequencing data and with lots of phenotypes. And we basically just asked them to run the same analysis on their own data. We send them the code and everything. And we got results that we think are roughly comparable for most of these phenotypes. And there are some notable differences which we think are interesting by themselves. So for example, you can see that in the Israeli cohort, the microbiome didn't really explain height. And in the Dutch cohort, it explained 26%, and it was really statistically significant. And that's interesting because we know that the Dutch people are the tallest people in the world. So I think we're onto something. So I don't know, maybe they have some superfood which causes them to be 8 feet tall and changes their microbiome. But you think? Yeah, but we didn't have access to this data. They ran the analysis. So I can't really look at their data and explain this, but this is what we got. Yeah. Yeah.
The risk factors you put into the model is compatible between the two cohorts.
Yes,
and the polygenic.
yes. That's a nice thing. That their study is very similar, so they had all these risk factors as well. Yeah, that was really cool that we were able to pull it off. It was really amazing. It was an interesting collaboration. All right. So in the short time left, but this is pretty short, I'm going to tie it all together. And so now we're going to try to predict phenotypes using all of these and see how much the gut microbiome contributes to the prediction. So I'm going back here to say a few words about how we do prediction in our statistical framework, because we're going to use exactly the same probabilistic framework. So again, we have these phenotypes and this normal distribution with the fixed effects, and it's exactly like before. But now we want to do predictions. So now we have this new individual here, and we don't know her phenotype. So we have the risk factors and the polygenic risk score, and we know the bacterial covariance between this individual and all the training set individuals and the environmental covariance. And so without going into the mathematical details, in order to do prediction, we just need to compute the expected value of this new phenotype conditional on everything that we know. So on the phenotypes, on the risk factors, the polygenic risk scores, and the matrices. So because we are using a very principled probabilistic framework, so we can do all of this in a very simple, straightforward manner. And so that's what we did. And again, this is pretty cool because this is a way to do prediction with millions of variables, millions of genes in our example, without running into all sorts of statistical problems, because it's all encoded in this covariance matrix. And for those of you who are interested, it's possible to show that this is actually equivalent to ridge regression that uses the principal components of this covariance matrix of the training set as features. It's not easy to, it's not difficult to show that. But this is, in a nutshell, how we did this prediction. All right, so what you can see here is a very rough explanation of our model. So we assume that the phenotype is given by the additive sum of age and gender and calories of different types of food, and the microbiome effect that is encoded through millions of genes, and the polygenic risk scores and some noise. And what we did is, so we did cross-validation, and we evaluated the ability to predict every phenotype using 4 different models. So the basic model just uses basic features like age and the age, gender, calories. But then on top of that, we added either microbiome or genetics and microbiome. So you can see here that the microbiome and genetics almost always contribute to the overall prediction. But it's more interesting actually to zoom in on the contribution to the prediction over this basic model. And what you can see here, which we think is really cool and interesting, is that the contribution of the joint model is basically almost always to the sum of the contributions of the 2 individual models that used only genetics or only microbiome. And we think that's cool because this is another indication that these effects are independent in some sense, meaning that the effect of microbiome is not mediated through genetics or the other way around, which we think is interesting. And yeah, I'm wrapping up. So in this part, we proposed 2 association measures between microbiome and phenotypes, which is microbiome association index and phenotype prediction. And we showed that microbiome association index levels are comparable to SNP heritability estimates, which we think is a big deal, because this means that microbiome composition really statistically explains a very significant fraction of many host phenotypes, which we are the first to show in a principled manner. And we showed that microbiome and genotypes contribute additively to predicting host phenotypes. And this, by the way, is a figure that the graphics department at the Weizmann Institute prepared because we submitted it as a potential cover for Nature. But apparently, it didn't get selected. But it's really cool, so I put it here. And I don't know why they didn't select it. I would definitely have selected it. Yeah. All right, so I want to thank our many collaborators, especially Daphna Rothschild and Elad Barkan, who are the other co-authors of this paper, and Eran Segal, and everybody else who collaborated with us. And thank you.